Biology Part 57. The tissue cells are removed from the gas exchange structure, so it becomes important to ask how gas exchange is controlled to meet the varying needs of the tissues. For example, during exercise, the muscles use oxygen and produce more carbon dioxide. This leads appropriately to an increase in both tidal volume and frequency of breathing. But how is this change in oxygen demand signaled to the respiratory system? You can understand some of the process involved by considering the flow diagram in the figure. The most vital part of this control system is the inspiratory center located in medulla. The medulla of the brain, the inspiratory center, consists of the nerve cells which generate periodic bursts of the nerve impulses. These impulses pass down the phrenic intercostal nerves, causing the contractions of the diaphragm and the external intercostal muscles, which bring about inspiration. Expiration, as you will recall, can occur passively, but in some cases it is positively assisted by the nerve impulses from the expiratory center, which cause contraction of the internal intercostal muscles. The inspiratory and expiratory nerve cells inhibit each other so that they cannot be active simultaneously together. The two centers are responsible for the basic rhythm of breathing movements. If the phrenic and intercostal nerves are cut, breathing stops immediately. The remaining elements of the control system act to modify the basic patterns of the nerve impulses produced by these centers. In this way, the respiratory system is able to respond appropriately to stimuli received by each of the following sense. Number 1. Stretch receptors. The walls of various air passages in the lungs contain numerous stretch receptors which become active during inspiration. Signals from the receptors help to terminate the burst of impulses from the inspiratory center and thus prevent overexpansion of the lungs. Number 2. Carotid and aortic bodies. These are located as shown in the figure. The carotid bodies are more important, but both types of structure contain specialized chemoreceptor cells. These cells are capable of responding to just two different stimuli. Firstly, they are sensitive to PO2, the partial pressure of oxygen dissolved in the blood plasma. Low levels of PO2 stimulate to the receptor cells and lead via the medulla to increased ventilation of the lungs. The second but most important stimulus for the chemoreceptor cells is PCO2, the partial pressure of carbon dioxide in plasma. Carbon dioxide dissolved in blood plasma to produce carbonic acid according to the following equation: CO2 plus H2O, reversible reaction, H2CO3, reversible reaction, HCO3, and plus H. From this, it follows whenever plasma CO2 is changed to H+, the hydrogen ion concentration changes in a precisely corresponding way, and it is in fact H+ that carotid chemoreceptors respond. Even slightly raised levels of H+ are very effective in stimulating receptor cells, and like low PO2 are followed by an increase in ventilation. It is important to note that because of the receptors are so sensitive to H+, the concentration of carbon dioxide in the blood becomes main factor controlling gas exchange.